In the model by Watson and Crick, there are two strands of DNA that make up the double helix. The double helix is held together through hydrogen bonds that are between base pairs / nitrogenous bases. Here's a simple illustration of the DNA model. The nitrogenous bases have specific pairs. Adenine and thymine are a pair, cytosine and guanine are another pair. The hydrogen bonds connect to the phosphate backbone, which provides structural support. Base stacking also stabilizes the structure. It creates an alignment with adjacent bases. The purines are adenine and guanine. They are the same in DNA as they are in RNA. The pyrimidines are cytosine, thymine, and uracil. In DNA, it's cytosine and thymine. In RNA, uracil replaces thymine. The numbers around the ribose structure are carbon atoms. There are 5 of them. The different numbers are significant because it allows us to label each of the groups among the structure. Ribose is different from deoxyribose because of a different hydroxyl group attached to the 2-carbon atom. Major grooves of DNA are present when the backbones are further apart. Minor grooves are when the backbones are closer together. DNA-handedness is left or right, but is typically right-handed. Right-handed DNA twists away clockwise. If it's the opposite direction, then it's left-handed. Anti-parallel strands means that the DNA strands are side by side, but are opposite in direction. This is determined by its polarity. DNA with an increased salt concentration can have few hydrogen bonds. This occurs even when the temperature exceeds the Tm, which is the melting temperature of DNA. An increase of salt correlates to an increase of Tm, which increases the stabilization of DNA.